Okay, so we just finished talking about the different appendages and layers that we can find existing around a bacterial cell. And we talked about the glycocalyx as the outermost layer, then the S layer if it's present, and then innermost to that would be the cell wall. So the cell wall confers a very rigid structure, and it serves as an extra layer of protection against the environment. Some walls allow the bacteria to evade a host's immune defenses, and sometimes they're used to help the bacterial cell to adhere to a host cell as well. Now, bacteria use peptidoglycan as the core component of their cell walls, and this is a mesh-like molecule that includes protein and sugar subunits. So that's what you're seeing over here on the right is your sugar and proteins that are part of that peptidoglycan. So we say it's a mesh-like molecule. So kind of think of it as like a chain link fence, right? So a chain link fence is very rigid, but it's also a little bit flexible at the same time because you can take that fencing and put it around a perimeter of your land and it conforms to the shape of your land. So it's a little bit like that with this peptidoglycan construction. So when we talk about antibiotics later on in the semester, we're going to talk about some antibiotics, how they interfere with the construction of this peptidoglycan molecule. So it's important to know that this is the main component of a bacterial cell wall. In general, the archaeans tend to exhibit a lot more diversity in their cell walls than bacteria do, but the archaeans lack peptidoglycan. Instead, they have one or more layers of what we call pseudopeptidoglycan. So it's not the same as peptidoglycan, though. So we can classify bacteria based upon the cell wall construction, and we've talked about the Gram stain technique before. And we said that this is a technique or staining procedure that allows us to detect the differences between what we call gram-positive and gram-negative bacteria. And the differences between these two types of bacteria has to do with how their cell walls are constructed. So we're showing gram-negative bacteria first. We see with the gram-negative bacteria, we have kind of this basement membrane here, the plasma membrane. And so all membranes are made up of phospholipids, and that's that round structure there with the two legs coming down. I always call it the egg on legs model because it looks like an egg with two legs. So it's a phospholipid bilayer, meaning two layers of phospholipids. And then in the middle, this area here is the peptidoglycan. And so that peptidoglycan is sandwiched in between one membrane and then a second membrane, which is this one here, which is the outer membrane. So it's almost like you're making a sandwich where the membranes are the bread and then your peptidoglycan layer is your meat or cheese or whatever you put in your sandwich. All right. So here it's talking about 1 to 3 nanometers in thickness for the peptidoglycan layer. So it's a very thin layer of peptidoglycan. If we compare this to our gram-positive cell walls, we see that gram-positive bacteria have a very thick peptidoglycan layer. So that's what we're looking at here. From here to here, this purple area is all peptidoglycan that's laying on top of the plasma membrane. So there's no outer membrane to gram-positive bacteria. We see that it also has this tectonic acid that's part of the cell wall construction. So this helps to stabilize the cell wall and also helps it to kind of maintain its shape, but it can also help it in some cases to bind to other cells. So if it's going to bind to a host cell, let's say. It can use that tecnoic acid to help with that. Now, notice that we're saying that it's 20 to 80 nanometers in thickness. That is the thickness of the peptidoglycan layer, so much thicker than that of our gram-negative. So you might think that gram-positive, because they have the thicker layer, that they're better protected from your immune system or from other organisms because of this thick layer. But in actuality, your gram-negative bacteria are the ones that are kind of better prepared to resist immune responses or to resist our antibiotics, let's say. And that's because if you think about it, they have the layer of peptidoglycan, but then they also have a cell membrane on top. So they have two different layers. So they have two different strategies that have to be overcome in order to kill the cell. Versus over here, we just kind of have this thick peptidoglycan, and then the plasma membrane is usually allows a lot of different material to kind of move through it. So your gram-negative bacteria are usually the ones that are better equipped to fight or are harder to kill. Now let's look at this in terms of our gram staining technique that we've talked about before. We mentioned with the gram stain you have a primary stain, which is your crystal violet. And so this is the very first step when you're trying to identify whether your culture has gram-positive or gram-negative bacteria. You would have the specimen placed on your slide, and you would add your primary stain of crystal violet. Now, if we have gram-positive, it's going to appear purple to start with. If it's gram-negative, it will also appear purple in color. And so this block here is showing the cell wall and how the composition of the cell wall affects our gram staining technique. So the gram-positive have that very thick peptidoglycan layer, which we said is kind of this meshwork, and the violet will be trapped in that mesh. Here we have the membrane that is on top of our peptidoglycan layer. So we have very little peptidoglycan for that violet to get stuck in, and a little bit might be attached to our membrane. Then we add the mordant, which is grams iodine. This doesn't change the color at all between our gram-positive and our gram-negative. It just binds to the crystal violet and makes these particles even bigger, so it's harder for them to be dislodged from that mesh-like layer. And we see kind of the same thing happening, right? Not so much though, because it's very hard to get to that peptidoglycan layer with the cell membrane on top. Now we get to our alcohol wash. So this is our decoloration stage or decolorizing agent. So it could be alcohol or alcohol and acetone. And now we start to see the difference between our gram-positive and gram-negative. So because the iodine acts as a mordant and helps to trap the crystal violet in the peptidoglycan layer of our gram-positive, they will remain purple. But any peptidoglycan that is, or excuse me, any crystal violet that is present is going to be rinsed off with the decolorizing agents. So it basically loosens up the cell membrane, so all of that gets washed out. Then we use our counter stain of safranin, and that will stain our gram-negatives, kind of a pink color that will then be retained, and that will not have any effect on our gram-positive bacteria. So this is how the construction of the cell wall affects our gram staining technique. Now there's one other thing that I want to mention with the gram-negative membrane. So this is kind of another view of our gram-negative cells, and purple being our peptidoglycan layer, once again thick for positive, thin for gram-negative, and the fact that we have this outer membrane for the gram-negative. Then we have these little green things sticking up off of the outer membrane. This is the type of glycolipid that's called a lipopolysaccharide, or sometimes they're just referred to as LPS, for lipid polysaccharide. So these are embedded in the outer membrane of our gram-negative. So you find these only in the gram-negative and not in your gram-positive. So this LPS actually contains, the lipid portion of it is kind of a toxin. And so this tends to make some of your gram-negatives a little bit more virulent and will cause disease symptoms because of the LPS that is present in the outer membrane. So we said that the archaeans do not have peptidoglycan in their cell wall. Instead, they have the pseudopeptidoglycan. So when we look at these non-typical cell walls, the archaeans definitely are not going to have cell wall construction that's like our regular bacteria. But we do have a few bacteria that have different cell walls. So when we were talking about staining tools and techniques, we talked about the acid-fast staining technique, and we said this works in lieu of our gram stain for bacteria that have very waxy cell walls. So at the time we had mentioned Mycobacterium. We said that this genus is responsible for tuberculosis and leprosy, and so this is kind of a non-typical cell wall, having that very waxy cell layer on the outside. Mycoplasma is also another genus that has some unique features, and what it is basically it lacks a cell wall. So these are very tiny bacteria, and they tend to reside within their host cell. So if you think about it, since they lack a cell wall entirely, they don't have any type of protection at all. So therefore they tend to reside within a host cell, which then protects them. If they are lacking a cell wall, then they just have this membrane around them, a plasma membrane, and therefore they tend to be much more pleomorphic compared to other bacteria. So walking pneumonia is an example of a mycoplasma. Right. So we've covered the cell wall in this little mini lecture. We've talked about the differences between our gram-positive and our gram-negative bacteria in terms of the construction of the cell wall. We've talked about bacteria having cell walls comprising of peptidoglycan, whereas archaeans do not have peptidoglycan. And then we've got these few other bacteria that have unique cell walls.